I had a lot of folks ask some really good questions out back. Some of them I want to touch on because I think those that were not related to personal circumstances may have been or are questions that some more of you may have, or when you hear the answer to may realize you had and didn't think of. And somewhat of an extension of our talk. Sometimes I can get up here and just talk, because there's a lot that I can say that an hour just doesn't afford me the opportunity to do. One of the questions that was posed to me is, how do I know when I go to my physician and I'm getting a diagnosis that it's what you called, quote, a comprehensive memory evaluation? So that's an excellent question, and it's one of my jobs is to educate you as health consumers on what you should have as an expectation when you go to your provider and say, I'm concerned about my memory and I'd like to have it evaluated. The first thing is, I mentioned that there are a variety of providers that are more than equipped to address this concern with you. And the manner in which they address it may reflect their specialty or their discipline. The kinds of disciplines that you will likely encounter, and I don't want to say in order of expertise, but what I will say is in order of breadth of service they would most likely be able to offer you themselves in their office. The first and foremost would be your family care provider or primary care provider. He or she would be the doctor you go to for your annual wellness exams, as well as the doctor that you may visit for, maybe even it's an internist, like an internal medicine doctor, for when you have traditional ailments like possible flu or strep throat or a virus or a bacterial infection. He or she is a perfectly appropriate person to address your initial concerns about your memory with. And if they are equipped in their office to begin the evaluation, I assume that they would. If they're not, they may refer you up the pipeline, so to speak, in terms of the disciplines that have more access to the sophisticated diagnostic tools we've discussed, like neuropsychological tests, neuroimaging studies, and things of that nature. We'll get there, I promise. But assuming you've gone to your primary care physician, it isn't sufficient to just get a health history, relay what your concerns are, and have he or she give you a diagnosis. All too often I see this happen where kind of promiscuous at the primary care level in prescribing Aricept. We're a little just overly enthusiastic about writing a prescription for Aricept. And while you may very well benefit from it because it is a global cognitive enhancer, it isn't necessarily equipping you with the knowledge of what that underlying health condition is giving rise to your symptoms. More importantly, if it is one of those differential diagnoses such as frontotemporal dementia versus Alzheimer, it's going to respond differently to donepezil and Namenda, importantly. And in some cases, if you have Lewy body disease, it's going to exacerbate your symptoms associated with aggressiveness and sort of gating. What we would say is appropriate conduct that we know is not appropriate in the restaurant, even if we see a nice looking waitress come by, making that kind of flirtatious comment or physically engaging with her. But if we have frontotemporal dementia, we may lack the ability to censor that activity. And that those symptoms are exacerbated by some of the symptomatic treatments for Alzheimer's disease. So having an accurate diagnosis is very important. And we shouldn't just submit to accepting the prescription for a cognitive enhancer based on exclusively an interview of medical history. The next thing that he or she is going to do, either in their office or refer you up, are neuropsychological tests. These are traditionally performed with pencil and paper, although some physicians are moving to computer-aided tests, and that is appropriate. If he or she can't do them in their office, they'll probably refer you to a neuropsychologist. This would be somebody or a specialized psychologist. This is likely somebody with a Ph.D. He or she is not going to prescribe you medications, but they are going to conduct the kinds of pencil and paper tests that are going to determine where the deficits are in those cognitive domains that Bernadette described. Processing speed, working memory, language fluency, delayed recall, all of that. They'll be able to query that, having you solve puzzles, having you describe relationships. Think of it kind of like if you've ever sat down and had an IQ test. There are some building exercises, there's drawing exercises, there's describing the relationships of how two things are similar or dissimilar. Holding information, doing mental math, those are all the kinds of assessments that they have access to using to help figure out where in the brain the deficit and where in the cognitive function the deficit lies. The next thing is, and a primary care physician can certainly do this as well and should as a first-line diagnostic approach, do a blood test to rule out nutritional deficits or vitamins and nutritional deficits, infection, systemic infection, thyroid or metabolic conditions. These are all kinds of things that can be determined by very general blood tests that they send off to LabCorp and do your traditional panels with. Sometimes it stops there. You know, if we've discovered that you have an underlying thyroid condition you didn't know about, or something along those lines, we can address it at the primary care level, and that's perfectly appropriate. That, along with the health history, they should be doing first. If that doesn't resolve a diagnosis, then they kick it up to the neuropsychological testing traditionally, along with an imaging study. There's two different kinds of imaging studies that we will request. The first is an anatomy study. It is a picture of the structure of the brain. It is achieved by MRI, and what we are looking for in this study is the volume of brain material that is there. So we take a picture of the brain and we take it through slices and we compose like a 3D image of the brain through those slices, and we measure the volume of the different brain structures. One in particular that's relevant for Alzheimer's disease is a structure deep. It's bilateral. There's two, one on each side, deep in the cortex, called the hippocampus. The hippocampus is very important for our ability to learn new information and remember it long term. And if you think about it, the way I always remember that the hippocampus is important for learning and memory is like if a hippo went to college, he would go to the hippocampus. So the hippocampus is the brain region that is paramount for learning and memory. I'll share a story of why that is in a second, how we came to know that medically. But we can measure the volume of the hippocampus by MRI and compare it in our patient imaging study to what the standard is for an age-matched healthy control. A male at the age of 72 who doesn't have a degenerative disease has a volume assessment of their hippocampus between a certain range. And if you are within that range, then we would assume you don't have degenerative conditions in your brain of the hippocampus. If you are outside that range, then we begin to zone in on what is causing the dementing condition. Is it a degenerative condition? The second kind of imaging study that we can perform is not anatomical, but rather metabolic. And this is called a PET scan. So this is an instrument that looks similar to an MRI. It's a big round tube that you lay in and we take pictures of your brain. This time we inject something in your bloodstream that travels to the brain. And there's two kinds of ways we can do this test. The one that Medicare reimburses for is called an FDG PET study. You can have it certainly done for brain activity. Some of you or your loved ones may have also had this test done for oncological purposes. We also diagnose cancer using an FDG PET scan. FDG is an acronym that stands for fluorodeoxyglucose. The word you probably heard, glucose, right? It's in our food, we eat it all the time, right? Glucose is what our body uses to make energy. It's what we use to fuel our cells, our muscles, and our brain. And in order to perform, we need energy. And in order for our brain to perform, it needs energy. So when we give our body this special glucose and the brain takes it up and breaks it down and makes energy out of it, the imaging instrument takes a picture of that and it's lit up brightly in the picture. It's because it has an emission spectrum that in the instrument we can cause to come out. And so the kind of image we're looking for in that case looks a lot like the radar map for Florida in June and July, right? In the wild thunderstorms with the high intensity are the red, orange, and yellow. And you know, we're not going to the beach that day. We're not playing pickleball, or we're not going out on the course that day. And the areas that are blue or green are the areas where we have sufficiently high pressure. We're not going to have a rainstorm. The image of an FDG PET scan is similar. Where there is the most activity and the most utilization of that glucose we provided, you're going to see hot. You're going to see red, orange, and yellow. And then the areas where you don't have very much activity taking place and little utilization of the glucose is going to look blue or green in the emission spectrum when we colorize it. And so the brain of somebody who has normal cognitive function is going to look like the Florida weather map. The brain of somebody who has Alzheimer's disease or other kind of dementing disease where they have less metabolism of the brain because there's less activity going on is going to be more blue or green looking. So we now know something about the anatomy and we know something about the metabolic activity. Again, have I said anything about plaque or tangles? No. So up until very recently, Alzheimer's disease, and still to some extent, Alzheimer's disease is a diagnosis of eliminating everything else possible. We didn't have a test that showed us plaque or tangle in the brain until we took the brain out of the skull and looked under a microscope. And you and I don't want that to happen while we're still living because we're going to be in trouble. So the diagnosis was always called probable Alzheimer's disease, and we went through all of those steps. And unfortunately, even our best experts were only about 60 or 70% of the time getting it right. In the process, they were identifying individuals who had encephalitis, hydrocephalus, all kinds of different things that could be causing dementia. And so it was important to go through the process. But even after all of these tests, we still couldn't confirm that it was actually Alzheimer's disease, because in order to have Alzheimer's, we have to have the presence of amyloid plaque and tau tangle. And none of those tests show with any resolution the presence of those two pathological features. 3 years ago, last June, so it'll be coming up on its 4th birthday, the Food and Drug Administration approved a new imaging agent. It's marketed under the name Amivid. And Lilly, I think, is the pharmaceutical company that has the rights to sell it. And it is an agent kind of like the fluorodeoxyglucose. When you inject it in the body, it goes through the body, and it's an antibody. It only binds to and sticks to amyloid. So it goes throughout your whole body, and it doesn't hang around anywhere that amyloid isn't present. Where the amyloid is present, it sticks. And then when the instrument causes, the PET stands for positron emission tomography. When it emits the wavelength that causes the special chemical group that we've added to the antibody to fluoresce or to emit its spectrum, then you get the same kind of heat map, only where the amyloid is present. So in this case, it's sort of the reverse. Now you see bright spots with the Alzheimer individual, and you see no yellow, red, or orange in the individual who doesn't have Alzheimer's disease, because the agent went all through the body and didn't have anywhere to stick to, so he or she just urinated it back out. Does it make sense? So now we know with certainty that amyloid is present. And we can even determine a percentage or a ratio of how much amyloid is present. So we know you are on the road to developing Alzheimer's and dementia symptoms. The problem is the one scan that just tells us about metabolic activity. Now it's important. And we try to parse out where it's happening. If you're only having reduced metabolic activity in the frontal lobes, then we say, okay, maybe it's frontotemporal dementia. And so then we'll do some more tests along those lines. If it's more widespread, it would be more consistent with Alzheimer's disease. If it's only in one small focal point, then it's probably the result of a stroke or mini TIA, several in one area. So again, it's kind of this deductive reasoning to get to a diagnosis, which a lot of medicine is, but it's not concrete. When on the other hand, we have this imaging test that is concrete. I can tell you with absolute certainty, the only thing that antibody is sticking to is amyloid. So if it's there on the image, it's because amyloid is there on the image. What do you think Medicare reimburses? The sensitive specific test or the global general metabolic test? Yeah, exactly. Your guess is in the first two don't count. Yes, it's the FDG scan. So the FDG scan costs about $1,200 in practice because it's $150 for us to buy the drug we inject you with, and it costs about $1,000 to run the instrument, pay the technologist, pay the radiologist to read it, etc. And Medicare reimburses, and you have your typical 20% copay. You know how much it costs us to buy the drug that binds to amyloid? $3,000 for one injection. $3,000, and we don't keep it in the refrigerator. We get it 2 hours before you arrive for your visit, and if you don't come, we eat there. It's $3,000. We can't inject it in somebody else. It is crazy. So now that cost winds up getting passed off to the patient. It's a $4,000 scam. So, but that's all supply and demand. If Medicare would reimburse for it, it would drive the demand up, and then they would back off that $3,000 supply cost. So I told you it was approved by the FDA 3 years ago, June. Now enough lobbyists have begged and pleaded with the Centers for Medicaid Services, CMS, to reconsider their position on amyloid scanning. Think about it. This is another biomarker. This is a screening tool that tells us about risk, that can tell us something about the disease development before dementia sets in. This pushes back the therapeutic window of when intervention may be successful. It's critical that we have access to this. It's no different than mammography or colonoscopy, right? If we treat heart, what good is diagnosing and treating after you have a heart attack? No, you take your Lipitor and your Crestor every day to day so you don't get the heart attack. This is where we are on the cusp of with Alzheimer's disease, but we need to be able to increase access to the biomarker screening technology of PET scanning. CMS is conducting an investigator trial. It's not the kind of thing where you put something experimental in your body and question whether or not it's going to change the outcome of your disease. This is a program where CMS is developing, it's called Continuing Evidence Development, to approve a treatment for a particular indication for reimbursement. So it's already using FDA-approved drugs and imaging modalities that are known to be safe and appropriate. And there are 18,000 participants available to do the IDEAS trial. And you have to be, you have to be 65 years or older. Your physician has to be approved to do the IDEAS trial, for which I think there are about 4 or 5 in the greater Tampa area. Our PET scanner is the only one approved to do the actual scanning, but you can still go to one of the other physicians. And I think you have to have straight Medicare If you have straight Medicare, it doesn't cost you anything. If you have a variety of Medicare Advantage programs, it may cost you up to about $200 to participate. And you will have a full physician diagnosis with all of the routine components I described, and you will also have an amyloid scan. And the point of the study is to understand from the physician's perspective, how did it impact their diagnostic decision-making and their treatment and management protocol? Did it improve their ability to differentially diagnose, and did it improve health outcomes? And so this is going to take a couple of years to conclude both the enrollment and the data evaluation. And then they will make a determination as to whether or not they will reimburse for amyloid scanning for the diagnosis, the differential diagnosis of Alzheimer's disease. So if that's something that you are interested in, again, we are doing IDEAS study patient scans. They do require you to work with our physician, because she's Amanda Smith and Christy Leach are both approved physicians to do this research. But you're more than welcome to get some information about that. Putting all of that together, that is what the comprehensive memory evaluation is. So after your neuropsychological tests, the individual who would interpret all of those imaging studies, the tests, the biological readout of your blood sample labs, would probably be a neurologist, a geriatrician, or a geriatric psychiatrist. They are best capable of interpreting that orchestra of medical data to determine whether or not your dementing disorder is Alzheimer's disease, frontotemporal disease, et cetera, et cetera. So that was a very long-winded explanation for how do I educate myself to be a good health consumer and know that I'm getting the most comprehensive diagnosis of my memory concern available. That was the long answer to that question. I feel like along the way, we also talked a little bit about clinical research. And what I didn't talk to you about is how do we conduct clinical research? I know you probably hear double-blind, placebo-controlled phase 3 trial. Like, what is all of that stuff? What does that mean? What is research that's valuable for me to participate in, and what isn't? This is really important because the media loves a good story. And the media really loves a good story that they can sell. And so when you find anecdotal evidence of things like coconut oil, who in this room isn't, quite honestly, isn't drinking, eating, or ingesting coconut milk in some form or fashion to improve the quality of their skin, their hair, their mental well-being, their cardiovascular health. You go down the list and you think coconut oil was the cure for everything, man. That was like the Ponce de Leon's fountain of youth. I put it on everything on my body just in case it works. Well, the problem with coconut oil is that there may be, indeed, and locally, anecdotal evidence of improvement. But as a physician or a researcher, we cannot advocate that because it's not evidence-based. It has not been subjected to the rigor of a double-blind, placebo-controlled trial. So when we take a product through clinical evaluation, it first starts out on, usually, I'm not gonna lie, as a happy accident in the laboratory in an animal model. We thought we were doing something, we spit in the vial, and somehow magic happens. I mean, most of the time, it's not the pursuit we were intending, it's not the hypothesis we had, it's usually an accident. And it's like, wow, wait, something worked. And so we chase that down, and we determine in preclinical drug evaluations, using animal models of disease, traditionally mice and rats, lower vertebrate organisms. We don't move up into primates very quickly. They're very, very expensive to do those studies, particularly aging studies. We have to age a primate 45 or 50 years before we can get an age-related result out of it. That is very expensive. We only have to age a mouse or a rat 18 months to get that same knowledge of the aging process, much better in terms of our pocketbooks. But we take the preclinical drug evaluation all the way through an animal model, and we demonstrate target specificity. We demonstrate how the body metabolizes whatever the agent is. We evaluate what the predicted outcome is going to be. If it's in a mouse, do they perform better on a learning and memory test? Yes, mice learn and they remember, and they can show us that on different tests, believe it or not. And once we have aggregated all of that data, we submit it to the Food and Drug Administration, and we ask for the right to administer a clinical trial to a human population. And the first phase, a phase 1 clinical trial, is where we take that drug and we take tens, like 10, 20, 30, multiples of tens of people who are healthy, do not have the disease for which the drug is intended, and we give them the drug. And we are looking for the safety and tolerability of that agent. And this is where you get all that fine print of like, the guy who talks really fast and low at the end of the commercial, may induce photosensitivity, that's where all that is sorted out. and so after the first couple of tens, this is what happens. We see the safety and tolerability of that drug in a healthy population. And assuming that it is safe and tolerable, it goes on to phase 2 clinical trial. Phase 2 clinical trial includes about 100 individuals, and this time it's in the diseased population. And so this time, if it's a drug against Alzheimer's disease, take 100 people who have Alzheimer's disease, and we're asking the same question: is it safe and is it tolerable? And if the answer is yes, then we start to say, what were the outcomes that were affected? We're not specifically looking for efficacy. We're looking for safety and tolerability. But it gives us a clue to how to design the next set of experiments for efficacy. Then we move to a stage 3 clinical trial, phase 3 clinical trial. This is where we introduce the double-blind placebo control. What that means is, if it's a pill, and it's a small blue pill, then the placebo control is a small blue pill. The physician who's giving it to you doesn't know which is which, and you taking it don't know whether you are taking the treatment arm of the drug or the placebo. So the person dispensing it and the person taking it don't know who they are. The only one who knows who they are is the clinical pharmacist who created the 2 different pills for you. So that's double blind. The placebo control is what I described. Everything looks, feels, tastes the same. One has the active ingredient, the other doesn't. That's a phase 3 clinical trial. In this case, we are simply looking at outcome. Did we affect an outcome measure? In the case of Alzheimer's disease, it may be performance on that mini mental status exam or some other appropriate test. So that's phase 3 clinical trial. When they move to a phase 4 clinical trial or an open label trial, that means that it's still placebo controlled, but traditionally it's already been shown to be effective, and they're just working out dose or just like mechanism of distribution. Like, is it better to inject it or whatever it might be, The open label means at the conclusion of the study, if you were not in the treatment arm and you were in the placebo arm, you're going to be given the drug for free for the same amount of time that your treatment arm counterparts had it before it either goes to market, or even if it's on market, but you won't have to pay for it. So that's what the open label means. So those are the various stages of the clinical trial. Right now, there are over 60 medications in phase 3 clinical trials targeting mild to moderate Alzheimer's. 60. Think about that. If each of those trials are recruiting tens of thousands of people, we could only fill like 1 or 2 trials with a population of 25,000 people who live in Sun City Center and Kings Point and Valencia Lake. This is why we have that clinical trial mobile unit, and we have to come and find the people. Because we need hundreds of thousands of people to be able to test these 60 drugs that are in phase 3 clinical trials. We don't just need those people to be willing, we need them to have the right diagnosis at the right time and have access to come down to the professionals that are conducting the research and blah, 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 blah. That's why we are hoping for that fleet of mobile units. I think I probably more than exhausted the extra time that Debbie gave me. I was pretending not to see the guy in the background doing his like jumping jacks.
Okay, so one more question for you about the clinical trials. When you said that you have to test so many, do you have any idea the percentage before you have a successful number? Like if you test 100, you may end up with how many that are qualified?
Oh, for screen failures you're describing. Okay, so what every part of the clinical trial that you participate in, any of those medical tests performed, they're always free of charge to you. Traditionally, your first encounter includes your informed consenting and then a screening activity. This is where you may fill the requirements or you may not pass the requirements of participating. You could screen fail for a number of reasons. Maybe the study requires a PET scan, but you have metal in your body and you can't take a PET scan, so you can't be part of the study. Or maybe you are required to not have cancer in the last 5 years with chemotherapy treatment, because we know that chemotherapy has effects on cognitive performance. They are short-term in the way of a couple of years, but nevertheless they could be confounding any results that we have. So those are the kinds of reasons that you may screen fail. We will often know on your first visit if you are going to meet or fail the eligibility criteria. It won't be weeks of investment on your part.
Okay, perfect. Thank you. And thank you for your
Yeah,
time.
no